Afternoon again. My talk will be on the suitability of the defoliating beetle, which is Physonota maculiventris, against the Tithonia diversifolia in South Africa. Introduction. The origin and global distribution of Tithonia diversifolia occurs in the tropicals and subtropicals areas of in Mexico, which is its native range. It has been observed and recorded and concluded as an aggressive weed in eastern areas of South Africa, which is Kwazulu-Natal, Limpopo and Mpumalanga. The uses of this weed: the leaf extract, it is used for malaria. It is used as a remedy for the malaria, intestinal parasites, and domestic animal skins. In some other parts of the countries, they use it to treat liver diseases and stomach aches. It has insecticidal properties, which is used against the termite infestation. Some other farmers in some other countries, they use it as a manure for their commercial and agricultural crops. This is basically the distribution map of the Tithonia diversifolia in South Africa, which is found in the KZN, Mpumalanga and Limpopo. Weed attributes: it is known to have allelopathic properties. It produces large number of seeds, which can spread a large number which can spread for a long distance and cover large part of the land. It prefers wide range of habitats. It occurs along the roadsides, river banks, and the abandoned farmers. It has long period of seed dormancy. Due to its allelopathic, it is able to get rid of the native indigenous plants. And in some other countries like Nigeria, they have reported that where it occurs, in Nigeria they have reported that if you can chop down this plant and introduce other plants like agricultural or commercial crops, those plants will produce small shoots and small roots due to its allelopathic properties. In other way, its allelopathic properties, its allelopathy, it can persist for a long period, even if it's not longer there. This is one of its attributes which facilitate its spread in the habitat. The humans and the wind are the major seed dispersal vectors which have been reported against the spread of this target weed, which is Tithonia diversifolia. This is basically the plant that I was just talking about. That's the flower, which carries large number of the seeds. That's the leaves, which are mostly 5 to 13 centimeter in size in most areas of the KZN and Limpopo, but it's not always like that. And it produces 3 to 5 lobes. This is the distribution of the weed. It occurs along the roadsides in Mpumalanga, and this one over here is KZN, and whereas that one is Mpumalanga. Here it invades the inlands, whereas in Mpumalanga it mostly prefers the roadsides. Control strategies: mechanical and control methods are expensive and unsuitable. Why mechanical is unsuitable against this weed? I have already indicated that T. diversifolia, it has allelopathic properties. These plants can manage to get rid of other native plants and create the space for its seeds to germinate. So when you cut this plant, the well-cut stem, most of them will just fall on the area which has been cleared by the allelopathic properties, so that when these stems falls, they will germinate. I'm trying to say the allelopathic of the plant it creates the microsize for the stem that falls on the ground. So when cutting the stems will just produce roots and germinate. Hence, they will increase the expansion of the weed. The biological control program was initiated by ARC-PPRI in 2007, and a number of the insects have been introduced in South Africa since 2007. A leaf defoliating tortoise beetle Physonota maculiventris is one of the selected and collected insects for the further screening for the host specificity. This is basically the map showing the areas where the insect it has been collected. It has been collected in various areas which are far from each other in the native ranges. And then this is the life cycle of the insect. They prefer the lobes, the tip lobes, but on the underneath of the leaf. This is how the egg batch it looks like. The eggs are just inside covered with this protective coat, and it functions again to protect the eggs against the predators. It normally carries up to 30 eggs, it normally carries up to 30 eggs. And this is how the larvae damage the leaves of the Tithonia diversifolia. This is how the pupa it looks like, and then the newly emerged adult. And then when it approaches the reproductive stage, it turns to golden brown. This is the comparison of the damages that are being done by the adults and the larval stage, and the larvas and different larval stages. The adults, they take everything on the leaf and the left the main ribs, whereas the larvae they just take the green part and leave the mainlets and the mid ribs hanging. When adults approach the reproductive stage, they lay their eggs, and then from the eggs it hatch. It takes some few days to hatch, and then this is the damage that they do on the target weight. The aim and objectives of the study is to determine the suitability of the Physonota maculiventris for release against Tithonia diversifolia, whereas the objectives will examine the various biological aspects of the Physonota maculiventris, which is pre-oviposition period and oviposition period, life stages, developmental periods, and to determine the host specificity, whereas again to determine the impact of the Physonota maculiventris on the target with Tithonia diversifolia. Materials and methods. These are the materials that I have done to study the biology of the Physonota maculiventris. 28 degrees Celsius and 22 temperatures day and night respectively were used, and the 75% humidity, 14 hours light of the day were also set for the study. All the biological studies were carried out on living plants. The reason I went for the living plants is that this type of the insect, their larval stages, they mainly depend on the compounds that are contained by the leaves of the host, of the plant host. They normally extract compounds and retain them and form a slime, which is a fluid which covers their bodies, and the fluid it facilitates their molting and the movement and the feeding. When they become dry, if you give them dry plants, they also become dry and struggle to molt and develop. So cut leaves, they don't work well for the developmental studies for these insects. The studies conducted included the number of larval stages, pre-oviposition period, fecundity, egg incubation period, and the generation period were determined. The feeding oviposition, this is basically the no-choice test that was being conducted against the selected insect, and the centrifugal phylogenetic method were used to select the test plants against the selected insects. The ornamental and the indigenous target weed were confined with the insect in cages for further screening for the host specificity. Six sex insect pairs were confined with the plant for the period of 20 days to determine the host specificity of the insect. 20 days were enough to screen the specificity of the insect. Most of the insects they normally die after 10 days or 5 days. Feeding damage, oviposition, newly emerged adults were monitored and recorded on each plant. This is the result for the biology of the Physonota maculiventris. It's just a summary. And then I went for the number of egg batches per female, oviposition period, incubation period, number of larvae that are produced per batch, larval period, pupation period, pre-oviposition period, larval instar, number of larval instar, male and female mass of the newly emerged adults, and the generation period. The parts that I have highlighted with red color, these are the most highly damaging stages. The larvae can take 14 to 20 days damaging the plant, whereas the adults can take up 22 to 24. I have already showed the comparison of the damages in the previous slides between the larval and the adults. So these are the days that they take they take in for the damage they take to damage the plant leaves. And if you can check here, the mass of the females exceed the mass of the males, and this really shows that the females they damage more than the males. Checking on the mass of the newly emerged adults in terms of the males and the females, and I went further and determined the sex ratio, the number of the females compared to the males that are contained in one egg batch, and then it showed that one egg batch it carries more females than males, which are highly damaging. And then this is the summary table of the test plants that I have tested. In total, there are 64. The insect it has rejected 62 plants, and then this 64 it includes the target weed together with the other test plants. And this one it is showing the plants that have been attacked by the insect. The first one is the target weed with Tithonia diversifolia. This is the damaging rate, which is normal feeding, and the insect has only shown the minor feeding on some cultivars of the sunflower, which is sunstripe, axan, and nongana. It failed to lay eggs on one of the three of the sunflower cultivars, whereas it laid some egg batches on the two cultivars of the sunflower. I haven't recorded any adults from this sunflower stripe, but the experiment is still going on. Whereas I have recorded 78 adults from the target weed in the same conditions. All these plants are in the same condition, but I haven't recorded any adults from the cultivars of the sunflower. Discussion and conclusion. The Physonota maculiventris, I have concluded that it has short relative generation period, therefore it has a potential to damage the target weed. Other than the target weed, the insect has only developed from the target weed. The rejection of the large number of the plant species, it shows that this insect is highly specific to the target weed. And the insect, I can conclude that the insect is still promising to be a biological agent for the Tithonia diversifolia, and it could reduce the threat that is being posed by this weed in South Africa. My future paths, will focus on the multi-choice test, impact studies, and the risk analysis. And these aspects will further give us indications of how suitable this insect it is and how it can manage in the field to reduce the threat which is being posed by this weed. And then acknowledgements are directed to working for water supervisors, David Melan, Terry Olkas, and technical support from ARC staff. Thank you.
Thank you very much, Tshililo. Any questions for Tshililo? Nothing. You've flabbergasted everybody.